I'm not going to ask you to know the various different groups within the bacteria and the archaea. You can give a big sigh of relief over that. In general, I'm not really interested in whether you know the names of all these groups or can memorize details about them. What I really want you to know is why they are important. And it is important for you to recognize certain groups within them, but I'm not going to ask you to memorize all of their diversity. So let's start off with some of the important roles that bacteria play. By far the most important roles that bacteria play in the world are ecological roles. The cyanobacteria, in particular, dominate a lot of aquatic environments, both saltwater and freshwater. They are photosynthetic, so they produce oxygen. They also produce a lot of nitrogen as a byproduct. And they generate a fair amount of biomass that then goes on to feed other organisms. The proteobacterium rhizobium is also incredibly ecologically important. This is one you've heard of already. These are the nitrogen-fixing bacteria that live in a mutualistic association with a lot of different plants, particularly those in the pea and bean family. When you think of bacteria, though, you probably don't think first about their role in ecology. You probably think first of their role in killing people. And there are a lot of pathogenic bacteria, some of which are extremely dangerous. A lot of the pathogenic bacteria are in the gram-positive group. There are things like anthrax, botulism, tetanus, gangrene, and hopefully the only one of these that you've actually had, strep throat. Extremely painful, but if you hit it with the right antibiotic, it goes away within something like 24 hours. The proteobacteria are another group of bacteria that contain a lot of harmful members. Cholera and dysentery are not diseases that you've probably experienced before, but they kill huge numbers of people worldwide in places where there is poverty and poor health care, and in particular, poor water treatment. Gonorrhea and Legionnaire's disease are bacterial infections due to members of the same group. We're not really familiar today with how important bacteria as pathogens have been during most of human history. It's not too much of a stretch to say that for much of human history, our primary opponent on the world stage was bacteria. You think of things like, say, the Black Death, which killed tremendous number of people across Europe. That was a bacterial disease. Today, something that you should be very much aware of every time you go out in the woods or the fields in spring or summer is Lyme disease, a fairly newly emerged spirochete bacteria that, if not treated, can leave you with a condition that will cripple you, potentially for the rest of your life. And finally, there are some diseases that you read about in, say, Victorian novels that we thought we'd left in the past, but that are coming back. Tuberculosis is one of them. This is the tuberculosis bacterium here, and the spotting that you can see on these x-rays is what happens when someone has a bad case of disease caused by this germ. Tuberculosis used to be extremely deadly. But frighteningly, it is making a comeback in parts of the globe, and it's coming back in the form of strains that are resistant to most of the antibiotics that we can throw at it. I'll say a little bit more in just a minute about why that is. I would be remiss if I didn't say that, frankly, there are some bacteria that are extremely useful to humans. Without including the vital members of your microbiome, which help you digest food and actually may help your body develop properly while you are in utero. There are a number of other bacteria that have proven to be very useful to us. Bacteria have been our constant companions since we started making food, and we found that in some cases they make food better. Examples of this are things like Swiss cheese and vinegar. And ironically, many antibiotics have been discovered that were generated by bacteria. The reason is that bacteria are often one another's most serious competitors. And so many bacteria have evolved chemical compounds that kill other bacteria. There's a tremendous amount of bacterial diversity that we don't yet know about. There's a tremendous amount of chemical diversity in those bacteria that we don't yet know about. And it's quite likely that the next big antibiotic will be something that has been discovered in some other form of life, maybe a bacterium. If you look at the phylogeny of bacteria and map certain traits onto it, you'll find that things start looking a little odd. So for example, these purple lines here, this is not an up-to-date phylogeny, but these purple lines here refer to photosynthetic capacity that is found in a variety of different groups of bacteria. It's not found within all of the low GC gram-positives or the alpha proteobacteria or the gamma proteobacteria, but it's found in some of those groups. And what's weird is it's the same kind of photosynthesis. It's the type of photosynthesis found in cyanobacteria, and a lot of the details are exactly the same. This is really weird because photosynthesis is an incredibly complicated mechanism. And if it were to evolve more than once, it's inconceivable that all of the details, or even many of the details, would evolve in the same way multiple times. So why is it that all of the photosynthetic bacteria don't cluster together when you look at a phylogenetic tree, when you look at a cladogram of them? The likely reason is that genes have not just been passed down from a common ancestor to all of its descendants. In some cases, it's very likely that genes have been swapped laterally, so that after two groups diverge, some of the genes over in this lineage moved over to this other lineage. This happens a disturbing amount in prokaryotic lineages. We tend to show you phylogenies that look like this, where you have this nice, neat progression. One lineage splits into two, that splits into another two, and they never ever rejoin. Unfortunately, particularly early on in life, and particularly among the prokaryotes, evolution seems to have looked a lot more like this. This is what's called reticulate evolution. It's evolution that looks like a net or a web, because a lot of genes have been transferred laterally among lineages. It's not this really clear branching. Now, this doesn't seem to happen, thankfully, in a lot of the higher sort of multicellular eukaryotes, things like animals and fungi and plants, although you do see it sometimes. It's mostly restricted to the single-celled organisms, but it makes a real mess out of reconstructing their evolutionary history. There are a few different ways that these single-celled organisms, and in particular bacteria and archaea, appear to be able to laterally transfer genes. In some cases, with some bacteria, there's this process called conjugation. Without going into the details of it, what happens is that a little tubule opens up and creates a passageway between two bacterial cells. And one of the accessory chromosomes is accessory circular chromosomes called plasmids. Inside one of those bacteria unspools itself, and a copy goes over into the second bacteria. In this case, this plasmid codes for the proteins and the processes necessary to make this tubule. So it basically passes on the ability to pass itself on. Okay, this is fairly organized, but there are also a lot of these prokaryotic organisms that can simply just pick up free DNA from their environment. If they encounter DNA that's just floating around, who knows where it came from, they can take it into their cell and integrate it into their genome rather than just, eating it. And in this way, if two organisms, even if they aren't closely related at all, if they share the same environment, they can swap DNA back and forth. This process by which genetic material can be swapped among cells, whether they're in the same species or whether they're genetically unrelated, is extremely important for the evolution of these groups. And it's proved a particular problem when it comes to antibiotic resistance. The reason is that if you have a bacterium that is resistant to, say, ampicillin, and you have a different bacterium that is resistant to penicillin, and they have no way of laterally transferring genes, then the ability to get one bacterium that is resistant to multiple drugs is going to be very low. All you have to do is hit your bacteria with a simple cocktail of the two drugs, and every bacterium is going to die. However, when they can start swapping genes around, some of the genes they're going to swap are going to be the ones that confer resistance to certain antibiotics. And that's exactly what has happened in many healthcare settings, is the use of antibiotics slowly selects for bacteria that are resistant to that particular antibiotic. Over here, maybe the use of a different antibiotic selects for bacteria that are resistant to that antibiotic. And then those two strains trade genes, and suddenly you've got a bug that is resistant to both antibiotics. Maybe it picks up a third gene from a different group, and now it's resistant to 3. It picks up a few more, and soon it's known as one of these multi-drug resistant bacteria, some of which are truly terrifying because they are very close to being resistant to all of the antibiotics that we currently have at our disposal. I'm not going to say too much about the important roles of the archaea. There's a tremendous amount that we don't yet know about these organisms. We really only learned they were there quite recently because of genetic sequencing. And the more kind of genetic prospecting we do, the more of them we find. It used to be thought that the archaea specialize in extreme environments, very, very salty environments, very acidic ones, very basic ones, and so on. It turns out they're actually just about everywhere. In kind of what we think of as ordinary, everyday environments, as well as these very strange ones. Nonetheless, they are remarkable in their ability to tolerate extreme environments. So, for example, these are salt ponds. Down here, they're used for basically you take seawater, you put it in these ponds, you let it dry out, and then you harvest the salt. This red color is an organism called Halobacterium, and they even got the name wrong. Everyone thought this was a bacteria for a very long time before they realized it was actually an archaean. There are archaeans that can live at a pH of 0 and others at a pH of 11.5. There's an incredible number of them actually in the deep ocean, where they seem to be the most common organism. There are some under the deep ocean that use the energy from high energy compounds released from undersea vents as their energy source. And sadly, these are also the organisms that are responsible for flatulence. They live in your gut and they make methane. Don't say you never learned anything in this class.